Good morning, students. Welcome to the biology class. We are continuing with our same chapter, life process. Yesterday we discussed about the various types of nutrition and we started with the plant nutrition, and today we will discuss the very important process, that is photosynthesis. What is photosynthesis? That is a process in which the green plants synthesize their own food in the presence of sunlight, chlorophyll, carbon dioxide and water. And during the photosynthesis, the following event takes place, and these events should be in a sequential order, that is one after another. The first event or the first step, that is absorption of light energy, that means your sun energy, by chlorophyll pigment, and conversion of light energy to chemical energy in the form of ATP and NADPH, that is adenosine triphosphate, that is a full form of your ATP. Second step is splitting of water molecules into hydrogen and oxygen in the presence of sunlight, and the splitting of water in the presence of sunlight, that is known as photolysis of water. Lysis means split, means breaking, and photo means in the presence of sunlight. And the third event is reduction of carbon dioxide to carbohydrate, that is a glucose or that is a food. So we are having a 4 raw materials and we are utilizing that 4 raw material. The first raw material, sunlight, second chlorophyll, and they are converting in the form of ATP and NADPH. The third one is water, which is being split into hydrogen and oxygen, and this oxygen is being released into the atmosphere, and the last CO2 is being reduced into the carbohydrate. So by this way, the complete equation of the photosynthesis takes place. So in this image you can see this is the light energy coming out from the sun, falling on the green parts of the plant, that is your leaf, and they are having the pigment, that is chlorophyll, which convert that particular light energy into the chemical energy, that is the sugar is being formed, that is the food, with the help of a CO2. Along with that, that H2O is being splitting into the O2, and this O2 is being released into the surrounding. In which way are the step of photosynthesis differ in the desert plants? So sometimes a question comes whether these steps follow immediately. The word is immediately in each and every plant. The answer is no, because in desert plants the steps will take one after another but not immediately. So in desert plants, take up of CO2 at the night, because stomatas they used to be open at night, prepare an intermediate molecule, that is your ATP, NADPH and your carbohydrates, and the intermediate molecule is acted upon by energy absorbed by the chlorophyll during the day to make the carbohydrate. So the steps are being followed one after another but not immediately. Few steps takes place at the night and the other steps takes place at the day, and this is possible only in the desert plants where the stomatas are being opened only at the night, not in the morning. Next, in the very important topic, that is a stomata, its structure and functions. So what are stomatas? These are the minute pores present on the surface of the leaf, and each stomata is composed of two bean-shaped or kidney-shaped cells which are being known as a guard cells. Example is dicot plants like pea and beans. In monocot plants like wheat, grasses, the guard cells are dumbbell shaped. The outer wall of guard cells are thin and the inner wall towards the stomatal pore are highly thickened. The guard cell possesses chloroplast and they regulate the opening and the closing of stomata. So in this image you can see, so this is a dicot plant in which you can see these are the kidney shaped structures, that is your guard cells. And this is the outer, which is the thin, and this is inner, that is the thicker one, and this white portion, that is your stomata or stomatal pore. So the guard cells, they contains the green dot, means they are having the chloroplast, and these are the subsidy cells through which the water is going to be taken in or being released out. So this is what called as the stomatal apparatus. So what is the difference between the dicot and the monocot plant? Only with the structure of the guard cell, means the shape of the guard cell. In the dicot, the shape of the guard cell is bean shaped, whereas in monocot the shape of the guard cell is dumbbell shape, जो exercise में use करते हैं जैसे. So these are the dumbbell shape in case of monocot and the kidney or the bean shape in the dicot. Function very important: exchange of gases, that is oxygen and the carbon dioxide, for the purpose of photosynthesis and respiration. Large amount of the water lost through these stomata in the form of water vapors known as the transpiration. So the stomatas, they plays a very important role in exchange of gases. Along with that, the loss of water, which is in the form of a water vapor, that is the transpiration, and performing are two very important functions. And next, and the very important question is, how do the guard cells regulate opening and the closing of the stomatal pores? The swelling of the guard cell due to the absorption of water causes the opening of the stomatal pores, while shrinking of the guard cells closes the pore. So opening and the closing of the stomata occur due to the turgor changes in the guard cell, means if that particular water is inside, they are going to be swell, that is called as a turgid, and if the water is being released out, they get shrink, that is called as a flaccid, means they are going to be closed. So when the guard cells are turgid, means swell, stomatal pore is open, while in the flaccid condition, that is shrinking, the aperture is closed. So as such, the question is very important, that how do the guard cells, they regulate the opening and the closing of the stomatal pores. In this picture you can see the guard cells, they are being swell up, they are having the water, so this is the water insides. So because of the swelling, because of the turgid, and that stomata is open, but here the water is being released out, so that causes shrinking, that is the flaccid, so the stoma is closed. So that depends upon the water intake. If it is absorbed by the guard cell, then it is open, and if it is released out, that becomes shrink, that becomes flaccid, so that cause the closing of the stoma. This is very important diagram you have to draw in your fair notebooks. Next is raw material required by the autotrophs for building their bodies. So in this plant nutrition, we discussed that the plant requires the four raw material, that is CO2, water, chlorophyll and sunlight, to carry out the photosynthesis. Whether the plant requires some another material, yes, they require nitrogen, they require phosphorus, iron, magnesium, which are being absorbed or which are being taken up from the soil while we are giving the fertilizers, manure to the soil during the plant growth, and they are essential, means nitrogen is an essential element used in the synthesis of protein and other compound, and nitrogen is absorbed from the soil in the form of nitrates and nitrites. You must have discussed, or you must be knowing about the nitrogen cycle in your 9th class, by which the nitrogen is being absorbed from the soil to the plants that is in the form of a nitrates and the nitrites with the help of a nitrogen fixing bacteria that is rhizobium. So in your NCRT we are having a two activities, that is 6.1 and 6.2. In activity 1, the aim of that activity is to demonstrate that chlorophyll is essential for the process of photosynthesis. For that they have taken the leaf which is the variegated leaf. Variegated leaf का मतलब है कि कहीं कहीं पे येलो पोर्शन है और कहाँ कहीं पे ग्रीन है. तो when you take that particular leaf, तो सबसे पहले इस particular procedure, how to follow the procedure, we take the plant and we have to keep that plant in the dark for 24 hours so that the complete leaf will be having a same amount of the photosynthesis. So after that you just keep that leaf or that plant in the sunlight for 6 hours and then pluck one leaf. For example, we have plucked that leaf and boil it in the alcohol so that the color of the leaf, that is the chlorophyll, is going to be dissolved and it will become a colorless. And after that we put the iodine, and if that gives the blue black color like this way and that is absent in this portion, means the green plant, the green part of the leaf which is having the chlorophyll is giving the positive test with the iodine, that means the starch is there, food is being synthesized only in the green part and not on the non-green part, so that will give a negative test. So what are the observation of that activity? The blue black color appears only in those parts of the leaf which are green in color, and variegated means that is the non-green part remains unaffected. Why? The starch formation takes place only in the green part of the leaf which contain the chlorophyll. Thus experiment shows chlorophyll is essential for the photosynthesis. The second activity is to demonstrate that CO2 is necessary for the process of photosynthesis, and to demonstrate that, to explain that what procedure we have to follow, that we have selected the two plants, same plants, kept it in the dark for 24 hours and then we kept them in the sunlight and surrounded by, means we have to make them airtight by the bell jars. In your plant A, we kept potassium hydroxide in watch glass and then make them airtight, whereas in the second we are having no potassium hydroxide, that is without, and this is with. So after 6 hours, what observation we will get, that if you pluck that leaf and boil it in the alcohol and then put the iodine, you will get a negative test, matlab no blue black color. But if we pluck that leaf, boil in the alcohol and then put the iodine, you will get a positive test, means that is a blue black, because photosynthesis takes place in the second part where the no potassium hydroxide is there, whereas it is negative in the plant A where the potassium hydroxide is present, because potassium hydroxide absorb all the CO2 present in the bell jar, and without CO2 plant cannot synthesize the starch, means no photosynthesis takes place. So observation, the leaf of plant B which does not contain KOH turns blue black, showing the presence of starch. The leaf of plant A which contains KOH remains colorless, showing the absence of starch. So what conclusion we get? Since the CO2 was absorbed by KOH in case of plant A, therefore photosynthesis does not occur and there is no positive starch test. Whereas the second one where that is not being absorbed, so this shows the positive test. So the normal photosynthesis takes place in plant B in the presence of CO2. Thus experiment shows CO2 is necessary for photosynthesis. So by this we finish about our plant nutrition, in which we discuss, that is, what is the autotrophic nutrition, what are the raw material utilized by the autotrophs, what are the sources of raw material, what is the site of photosynthesis, what is photosynthesis, what is the equation of photosynthesis, the steps followed in the photosynthesis. Then we discuss about the stomata, the structure of stomata, the function of stomata, opening and the closing of stomatas, the raw material required other than CO2, water, sunlight and chlorophyll, and the activities which shows the chlorophyll is necessary for photosynthesis, CO2 is necessary for photosynthesis. These all are very important. So that finishes your plant nutrition. So please go through the in-text question of NCERT. I try to give the answer of each and every question. But please go through it. If any query, you can contact me. Till then, stay home, stay healthy, stay safe. Thank you, and our next lesson will be on animal nutrition.